Hello guys, welcome to my channel. As you might all know, I used to do covers here. I sing, but since it's time for online learning, I'm going to use my channel for my lectures. So aside from being an artist, I'm also a doctor, a lecturer in MHAM College of Medicine in Cebu City, Philippines. So without further ado, I'm going to present my lecture on the genome. So let's start. Our textbook for pathology is the Robbins and Cotran Pathologic Basis of Disease. And now it has a 10th edition. And if you have this one, the ninth edition, you can still use this one. Actually, I've browsed through the book, and you can also download a PDF form. There's not much difference except for the topic on COVID. Okay, you can also use this handy reviewer on. Actually, it's a USMLE First Aid Reviewer, which summarizes the important topics in each subject. Okay, not only in pathology but for the other subjects. Okay, it's a good reference if you are looking for a reference that will summarize one chapter in a page. Okay, so that can help you. And other references or other sources, you can look for um, BRS pathology. Okay, it's a reviewer that outlines some basic stuff in in a particular chapter. Okay, so if you're looking for a um, something that can help you outline the chapter, then BRS pathology can help you. Okay, so our lesson for today is on the genome. Okay, but uh, different from different in contrast to what we really do in class, which we really explain everything into detail. Okay, let me just outline, okay, the chapter for the benefit of your independent study. So I will not go into the details because I might get very boring. Okay, so the platform used, as I told some of your presidents, is Google Classroom. So for this chapter, for this lesson, I'm going to use Google Classroom. I do not know if I'll be able to stream this live via Zoom on your specific schedules, but you can always play this video. I'll paste a link of this lecture in Google Classroom. Okay, on the day uh, when you have your lecture schedule. Okay, so let me just remind everyone. Okay, for section BD, you are having your online lecture schedule on August 18. That is Tuesday. The lecturette starts at 8 a.m. There will be a short quiz at 10 a.m. and I will give you an assignment. The deadline will be at 12 noon. Okay? AC, August 20, that is Thursday, 8 a.m. That will be the time for the lecture. Short quiz at 10 a.m. and the deadline for the assignment is at 12 noon. EF, August 20 also, and that will be in the afternoon, 1 p.m. Short quiz at 3 p.m. and the deadline for the assignment is at 5 p.m. So I don't make much demand from you in terms of other requirements, but please make good in the unit exam. Okay, I'm referring to the unit exam that we will have for all sections on August 26 on Wednesday. 7 to 8 a.m. Okay, and in Google Classroom. So any questions? By the way, if you have any questions, please type it in the Google Classroom. Okay, so I'll try as much as possible to answer those questions. Okay. The goals for discussing this chapter is to discuss the important concepts about the genome, about the Human Genome Project, and also we'll be able to mention about the importance of the mapping of the human genome. Our previous knowledge on the DNA is that the DNA, okay, actually from macroscopic to microscopic, you have is contained in the chromosomes. Okay, remember that in biochemistry. And for each chromosome, it is composed of chromatids. And if you unwind them further, you see a densely packed inactive heterochromatin. And as we further unwind, okay, when it's relaxed, it's in the form of a euchromatin, and it's in the active form. And in the euchromatin form, we'll be able to appreciate the nucleosome. Okay, the nucleosome is a unit which is composed of the histone, where the DNA winds around, okay, and the DNA itself. And if we untangle it further, we can appreciate the basic unit of genetics, which is the DNA. The central dogma of genetics states that DNA can be replicated by itself, okay, into another DNA, or it can be also be transcribed into an RNA. And we know what's the purpose of RNA being transcribed. Okay? After all the splicing, removing the non-coding, non-protein coding parts of the RNA, this RNA is translated into amino acids which form the protein. Protein in the form of the cytoskeleton, protein in the form of the enzymes, and other forms of protein. Okay? In our previous knowledge of the DNA is that every DNA, okay, is being transcribed, and every RNA is being translated into proteins. Actually, that's true. Okay? 1.5% of all the genes are what we call as the protein coding genes. The total number of genes is 3.2 billion base pairs. Okay? So out of those 3.2 billion base pairs, you see you have 1.5%, which is 20,000 genes are responsible for protein synthesis. And the rest, which is the 98.5%, is involved in gene regulation. So so that means not all DNA is converted into proteins. Okay? Majority of the genes are involved in gene regulation. So those are non-coding DNA, the non-coding RNA, and epigenetics. So let's look back into the previous diagram. Okay. So which parts here are the non-coding? Let's see. The centromere is a non-coding DNA. The centromere, the part that holds two chromatid strands together, okay? The telomeres at the end, okay? which are responsible for the aging, and this one, this is an RNA, but then you see the promoter, you see an enhancer. These are non-coding RNA. You see the histones, okay? You can also see some modifications of the histones. So those are epigenetics. So let's go to non-coding DNA. So we'll be talking about genetic polymorphisms. These are variations in the DNA in the form of a variation in a single nucleotide. Okay, and that is what we call as SNPs. Okay, SNPs can cause a disease, directly cause a disease, or it can be a neutral variant. That means it doesn't cause a disease, but by itself, but it can cause a disease if it's paired or if it's in close proximity with another SNP that is causing a disease. Okay, the concept that I just mentioned is what we call a linkage disequilibrium. Kindly read that in the book. The second genetic polymorphisms is polymorphisms in the form of multiple nucleotide polymorphisms, or what we call as copy number variations. It is in the form of long stretches of DNA. Other forms of non-coding DNA are promoters, enhancers, silencers, and binding sites in the DNA. You also mentioned about the telomeres and centromeres. Transposons, if you remember your biochem, in the non-coding RNA, okay, we have two forms. We have two forms of non-coding RNA. That is what we call as the microRNA, which is involved in post-transcriptional silencing. Okay, so it is an RNA that is not involved in translation into protein. It remains as an RNA which is involved in the silencing into silencing of translation into protein. Okay, or we can also have a synthetically produced microRNA, what we call as the small interfering RNA. Okay, it is used in a knockdown technology. Okay, so for example, in the lab, we want to produce a rat which doesn't have any hair. We use this knockdown technology just to remove the RNA that is translated into protein, into the hair protein. Another form of non-coding RNA is what we call the long non-coding RNA. It's very much similar to microRNA, but the functions are varied. Okay, it can be used to activate a gene, to suppress a gene, modify chromatin, and assemble protein complexes. These all non-coding RNA are involved in the control of different diseases, varying from atherosclerosis to cancer. So this is a figure illustrating to us the concept of microRNA. Take note that we see two DNAs here. Okay, this one, actually two genes, and another one here. So this is a DNA, okay, which gets transcribed into an RNA, and later it wants to get to be translated into a protein. That's the plan, okay? Transcription and translation into protein. And another gene here, if you take note, this is what we call as the microRNA gene, okay, which is of course it's destined to become a microRNA. Okay, we have a primary RNA here. Okay, becomes a pre-RNA. Okay, and through this dicer or an enzyme called dicer, it becomes spliced into what we call as the mature microRNA. Okay, so how does this microRNA become responsible in transcriptional silencing? So look, this microRNA binds to a complex, what we call as the RNA-induced silencing complex. Okay, it acts very much like a transcription factor or a or a polymerase, but then it's involved in, of course, silencing. Okay, how does it silence a an RNA that is destined to become a protein, to be translated into protein? Two ways. You have, okay, translational repression, wherein this the risk complex, by the way, okay, binds with the microRNA and it blocks translation. Another thing is that the complex binds into the RNA and causes cleavage. Okay, it breaks the RNA that is supposed to be translated into protein. And the process is what we call as post-transcriptional silencing, or what we call as gene silencing. Okay, this figure talks about the functions of the long non-coding RNA. So it's long because it is composed of not just one single nucleotide, but it's, you see, you have a lot of nucleotide, okay, linked together. So this long non-coding RNA binds to a ribonucleoprotein transcription complex. Okay, remember, very much similar to the RISC complex here, the previous figure. Okay, however, in this context, this is involved. This complex is involved in transcription, in gene activation. Okay, so that means when the gene is activated, you see transcription and translation will proceed. Okay, it can also be involved in gene suppression, where this long non-coding RNA binds to the said complex, okay, and will not proceed to transcription and translation. It can also promote chromatin modification, wherein the binding of the long non-coding RNA and the transcription complex causes changes in the histones, causes what we call as modifications of the histones, methylation and acetylation. We will talk about that later. Next is that this complex, okay, becomes an attachment of other transcription factors, causing changes in the DNA. So we're done with non-coding DNA and non-coding RNA. Let's go to epigenetics. Epigenetics are stuff or are elements that are outside the DNA, and we are talking about the histones and the histone modifying factors, okay, or what we call as the chromatin complexes. Histones are protein, okay, protein that are where the DNA winds around, okay, causing a clumped DNA, especially in the inactive form. Okay, histone modifying factors are um, are f- um, histone modifying factors are methyl groups or acetyl groups or phosphates that bind with the histone, modifying its action. Okay, so and the following are types of histone modifying factors: histone remodeling factors or remodeling complexes, wherein it modifies the conformation of the DNA. Next is the uh, writers, okay, or the marks, okay, wherein, for example, if a histone is attached with a methyl group, it causes the silencing of that histone. Therefore, there will be no transcription, there will be no translation that will occur. Next is the histone acetylation, where this acetyl group activates. That means it causes some transcription and translation later. Phosphate group that attaches to the histone either can cause an activation or an inactivation. When a DNA, the methyl group attaches itself to the DNA, it causes an irreversible silencing. And when you have chromatin organizing factors as your histone modifying factors, it causes the looping of DNA, okay, thereby regulating spatial relationships between promoters and enhancers. We also have erasers and readers, okay. And why is epigenetics important? Epigenetics are important because these are when they are dysregulated, when there is disturbance in the epigenome, it causes diseases. Okay, it has a central role in cancer and in other diseases. So this is a histone, and when we want to count the units, you see you have 1, H1, you have H2, 2 units of H2, you have H3 and H4. Okay? And if we total them all, you see H1, 1, 2, 3, 4, 5. So you see, 6, 7, 8, 9. So you have 9 units of histones. H1 being the linker histones. There are 2 turns of DNA around a histone, approximately 2. So particularly you see you have 1.8 turns. The DNA, when stretched, DNA from one cell when stretched can be as long as 2 meters. Okay, but if it's clumped in a in a nucleus, it can be as small as 8 micrometers. So this figure illustrates the 2 types of chromatin: heterochromatin, which is in the inactive form, and the euchromatin, which is in the active form. The heterochromatin, okay, is cannot be translated because it is in the clump form, and these are what maintains it in the clump form. These are what we, if you take note, you have lots of methyl groups here in the histone. And in the euchromatin, you see lots of acetyl groups here, the yellow one. Okay, that's why promoting relaxation of the DNA. So how do we apply the concept of epigenetics into real life? Okay, so remember about the debate between which gives more weight in the development of a person. If you have is it the genes or the environment? Is it nature or is it nurture? So this is the answer. It's both. Both genetics and epigenetics play a great role in the development of a person. Okay? So genetics, that means that the DNA of a person. The environment also plays a big role. Okay? Environment or epigenetics. So for example, the person might have good genes. However, these genes are being affected by vices, by medications, and traumas, stress. Okay, it can lead to a deterioration of the DNA. This can also explain why twins, okay, monozygotic twins, differ from each other when they grow up because of environmental differences. Let me just explain to you a little bit about the concept on gene editing. Okay, it is based basically on how a certain bacteria um, fights its enemy. So in the book it mentioned about phages or viruses. What it does is that it takes the DNA of its of the previous enemy or the previous virus that it encounters, and it incorporates the DNA of that enemy into its own in the form of clustered interspersed short palindromic repeats. Okay, so it has the CRISPR into its own DNA, okay, and it's being transcribed into a guide RNA, and this guide RNA becomes bound into with a protein called a Cas9 nuclease, and this complex is the one that is responsible in fighting the next virus that it encounters. So, for example, this protein, okay, cleaves the DNA of the virus, okay, and causes mutation. Two mutations in the enemy's DNA. You have the non-homologous end joining mechanism, which is error prone, or homologous DNA recombination. So class, you might wonder uh, how this concept in gene editing among bacteria becomes applicable in humans. So imagine this is the RNA, the RNA of a certain cell without disease. Okay, this is a protein where it binds to. Okay, and this is the DNA of a cell, DNA of a cell, a certain segment of which contains the DNA that will be coded for cancer or diabetes. This portion here undergoes mutation, and therefore it doesn't become a diabetic cell or a cell that will be transformed into cancer. Okay, assignment. So the deadline of this assignment is stated below. Okay, for section BD, it's on it's August 18, 12 noon. AC, August 20, 12 noon. Section EF, August 20, 5 p.m. So very simple. The instruction is just tabulate any of the elements in gene regulation, just five, and corresponding to each, please write the application of knowing these elements, the application in science. Okay, so why is it important? And I will be giving a quiz. See you in Google Classroom, Section BD, August 18, 10 a.m. Section AC, August 20 at 10 a.m. Um, Section EF, August 20 at 3 p.m. So reminder, all sections, the unit exam for the topic on genome is on August 26. That will be Wednesday, 7 to 8 a.m. See you in Google Classroom. Thank you very much for listening.